Okay, the final speaker of this session is Dr. Javed Khan from the National Institutes of Health, who will tell us about genomics to identify novel biomarkers and drivers to enable precision therapeutics. Javed?
Thank you very much. Can you hear the microphone on? Can you hear me?
We now have a second microphone, so we're going to use this for questions from the audience.
So I can actually walk around, which is good.
Yeah. Well, I'll take this for the questions.
So I want to also thank Jeff and the organizers for inviting me. Actually, several years ago, probably about 15 years or so, we got funded by CCF, which helped us to launch a very nice database in terms of putting all our genomics data, and we're still actually using that to this day. So thank you very much for that. And Jeff also loved this idea, and I think I've been in this area for 20 years. I don't think we've had many of these meetings where sort of the multiple institutes that are doing research in pediatric cancer are actually getting together. And definitely, if you need volunteers, there'll be people at NIH. So I hope you do continue to do this. So thanks. All right. So also, a lot of echo here. So can you just turn me down a little bit? Yeah. Thank you. So Jeff asked me not to make this too technical, but I'm just interested. How many people are actually doing research at the moment in cancer research? I think quite a few of you, but not. So some are not. Okay. So I'll try to explain some of my slides as I go along as well. So the title of my talk is our lab does cancer genomics, look at the genome of cancers, and really we mainly want to find these new biomarkers, which are like markers for disease or response, and drivers, which are the genes that actually drive these cells to become cancer. And the idea behind it is, you've heard of the phrase precision therapeutics. That means that you precisely target the protein or what's wrong with the individual cancers. So that's the theme of my talk. So if you've heard this theme, from Jeff's talk an hour ago or so, that the childhood cancer therapy really has been the beginning of a really modern success story. You've already heard how from the 1960s to 1990s, the survival rate has significantly increased from a few percentage to over 80% in many diseases. And I've just shown here leukemia, lymphoma, and Wilms tumors. Really, the success has been attributed to very careful, Jeff presented earlier that there have been very careful empirical trials where basically you use a therapy works, you then improve on that therapy by adding more drugs to it, and you do clinical trials based on your previous observations. But the main problem nowadays is really metastatic disease and certain other cancers. So you can see here, what I've shown here is the brainstem gliomas, and you'll hear about some of these, I think, later on today. But many cancers still remain incurable, and brainstem glioma is one of them. But also, you've heard about metastatic cancers. If the cancer spreads from the site of origin, then still over the last 20 years or so, the survival rate hasn't improved and is still less than 30%. So this is true for Ewing sarcoma. You've already seen this comparing local disease versus metastatic disease. Rhabdomyosarcoma is what I'm talking about, and it's similar for rhabdomyosarcoma. So metastatic, for localized disease is curable, and metastatic disease is essentially incurable. And what I'm going to argue with you, or what I'm going to discuss with you today, is that the reason for this is the biology of these cancers, obviously. It's not just the therapeutics, it's actually the biology of these metastatic disease compared to localized disease is fundamentally different. And so I think similar approaches, exactly the same approaches cannot be done with these cancers. So what about, even though we've heard that there is a significant cure rate, for example, 80% or so, and this is rhabdomyosarcoma, non-metastatic, although there is a significant cure rate, it is at a significant cost. So here I've shown you some of the complications as published in this manuscript here, that there's certainly a significant amount of life-threatening complications of chemotherapy and other therapies, severe and even fatal complications. So there's certainly a cure is at a cost. So my lab essentially works on very high throughput data, so intense data processing. So essentially, this you know about how information is passed from the nucleus down all the way down to the protein level, going from DNA to messenger RNA to protein. And there's a whole bunch of loops in there where different components, proteins control mRNA, DNA, and then microRNA controlling transcription, translation, and so on. So really, the Human Genome Project, which began more than 15 years ago, wanted to basically catalog the entire human genome. And it started off with actually sequencing the human genome. So all the DNA, the letters of the nucleus in your DNA, the 3 billion of these letters have been essentially sequenced for humans and multiple other species. And then from that emanated many of the other methods to find out what are the other components of the genome. And these include messenger RNAs. And I've just shown here how many thousands of genes there are. So there are predicted to be 60 or so thousand genes, 198,000 transcripts. 19,000 of these genes are actually protein coding genes. And with alternate splicing, there's at least 80,000 different proteins that are being made. There are a whole bunch of non-coding RNAs. I'm just showing some of the long non-coding RNAs, 15,000, and then small non-coding RNAs, and then more than 100,000 proteins. So huge amounts of data. And then just recently, which I won't talk about, but my lab is certainly working on the whole realm of epigenetics, how not the DNA itself, but actually the control of the DNA, termed epigenetics, actually also plays in the biology of these cancers. So when you put all this together, you find out that cancer isn't just driven by a single gene, but the whole process working together to determine how these cells actually behave, the phenotype. And this also determines the cancer that you have, the cancer diagnosis, finding some of the targets, and then also the response to therapy is dependent on the information that's actually coded within your genome. So my mission statement is a bit more complicated than yours in To Cure Cancer, which is sort of related. But essentially our group wants to harness the power of these very high throughput methods like genomics or proteomics. And at the NCI, we primarily see patients that have very high-risk disease. These are diseases that have spread or resistant to therapy or come back, recurrent diseases. And so our goals are to use these methods to look at the biology of these cancers, to find these drug targets, the targets for therapy, and then develop novel therapeutics. So I'm going to just show you a few stories based on using these methods on some of the cancers we work on. So I'm going to talk to you about how we use first-generation genomics to identify this gene called FGFR4, and you'll hear a lot about this gene in my talk. How after first-generation genomics, next-generation genomics comes along, and this is I'll show you a few slides on how powerful this technique is in looking at the genomics of cancers. I'm going to focus on rhabdomyosarcoma. I'm going to talk a little bit about the NCI's plan for precision therapies, so using genomics to guide therapies within the Center for Cancer Research. And at the very end, talk about immunogenomics. Hopefully it will pave the way for the next session in terms of how we're using immunology to develop therapies for pediatric cancer. And again, I'm going to focus on this protein, FGFR4. So just one slide of rhabdomyosarcoma. You've already heard a little bit earlier about Tanner. These pediatric cancers, these rhabdomyosarcomas are in fact divided into two subtypes. You heard about alveolar rhabdomyosarcoma, but also there's an embryonal rhabdomyosarcoma. And previous work has showed that some of the molecular changes include loss of heterozygosity of chromosome 11p in embryonal rhabdos, and then a 213 translocation in alveolar rhabdomyosarcoma, where chromosome 2 and 13 break and then join each other, producing a novel protein, which I'll talk about later. And you can see that the prognosis is very different where the patient has alveolar rhabdomyosarcoma versus embryonal rhabdomyosarcoma. The histology, they call alveolar partly in terms of the histology, it looks like a lung. It's like an alveolus of the lung, and they have small round blue cell tumors. So several years ago, Fred Barr, who is actually currently at the NCI, discovered that the 213 translocation results in the fusion of a gene called PAX3 with another gene called FOXO1, producing a very novel fusion gene, putting two proteins together that's never been seen in a cell before. And this, I'll show you, is a primary driver in these alveolar rhabdomyosarcomas. So again, many years ago, when I was in the NHGRI, in the really early days of genomics, we used a technique called microarray techniques to look at the gene expression levels in different types of cancers. What I show here is 4 different cancers: Ewing sarcomas, neuroblastoma, rhabdo, and lymphoma. And for those who aren't familiar, these green and red squares imply the level of expression of a particular gene shown here in a particular cancer. So the redder the box is, the more highly the gene is expressed. And what we found was that coded within the mRNA is a diagnostic signature. So each of these cancers have a specific gene expression signatures that actually tell you what the diagnosis of these in different cancers are. And I show you here how at that time we discovered that FGFR4 was actually very highly expressed in rhabdomyosarcoma. So it's just a blow up here. So within this list of genes, there are many drivers of each individual cancers, which obviously a single lab can't work on, and our lab is focused on FGFR4. So let me tell you a bit more about FGFR4. Why does my lab focus on this gene? So this is on the cell surface. It's a tyrosine kinase. It's an enzyme that produces the transfer signal from the outside to the inside of the cell. It's expressed in almost 100% of all muscle cancers. It's a direct target of this fusion gene I've talked to you about. So in other words, this fusion gene directly activates or increases the expression of this. It's important in muscle development, so likely to be involved in rhabdomyosarcoma biology, because rhabdomyosarcoma come from muscles, as you know. When muscles damaged and the muscles regenerate, this gene is induced during the regeneration phase and is not expressed in mature muscles. So all these indicated to us that this was probably a key target in these muscle cancers, so a possible oncogenic role. So we went on then to do essentially early or first generation genomics, which includes sequencing of this gene using Sanger sequencing. And what we found was that about 7.5% of these rhabdos essentially had a mutation in the enzymatic domain, which meant that these mutations essentially makes the gene continuously on. So it has high activity, resulting in increased growth, transformation to making cells into more cancerous, and then also increased lung metastases. And then when we used an inhibitor of FGFR4, we found that the cells that had the mutations were more sensitive. So that told us that these cancers were addicted to FGFR4, such that if we inhibited, these cells rapidly die. So that gave us actually important clues why, at least molecularly, we can potentially use this to target rhabdomyosarcoma. So, but I told you, so this is one of the problems of using these old types of sequencing methods, is that you can only do one gene at a time, and I show you only less than 10% of these rhabdos actually have the mutations in the gene. So that led us to the question: what about the other driver oncogenes or driver mutations? So then came along, fortunately, the technology called next generation sequencing, which some of you may or may not have heard of. Essentially, it's like Sanger sequencing, which is basically reading all the bases of the letters of the human genome, but not doing it one at a time, but taking every single piece of the DNA or the RNA, fragmenting it into billions of pieces, attaching sequencing like oligos, and then just sequencing each one of these fragments, and then lining up to the reference genome. So that gives you billions and billions of reads, and then you can tell then where each of these fragments actually come from in a particular tissue. So this is next generation sequencing. And so this is a slide, a cartoon I took from Matt Meyerson's talk, but you can see the power of this technique. So what I show you here on the top is a reference human genome, and underneath are the sequences. There's all those fragments that you break apart and sequence. And the color, the orange, tells you that we sequence the ends and align them to the genome. So that tells you immediately that in the patient's tumor, for example, instead of an A, there's a C. So this is a point mutation. Sometimes we see pieces of DNA missing. This is an insertion or deletion. Sometimes a whole segment is missing. So it tells you there's a deletion of both copies of this homozygous deletion. Or sometimes it's only half, only one chromosome is missing this part. Or sometimes there's amplifications, which means more copies. Or sometimes there's a fusion gene between two chromosomes, or even some non-human sequence. So you can see here how powerful this method is as a single methodology to interrogate the genome of cancer. But not only DNA sequencing, but RNA sequencing is an incredibly powerful tool as well. So, in RNA sequencing, essentially you take the RNA fragments and align it to the human genome, and it gives you an exact digital gene expression profile. It tells you, for example, if a gene that's mutated in the DNA is also in the mRNA, hence the protein is actually produced. So this is called an expressed somatic mutation that occurs in the cancer. It gives you information about alternate splicing. It gives you information on if a fusion gene is actually expressed or not. It tells you potentially immune signatures within the cancer and all sorts of other things, including RNA editing, novel transcripts, and non-coding RNAs. So a very powerful technique that has replaced microarrays. So with that background, we essentially went on to, again, look at rhabdomyosarcoma at much higher detail, and we had a discovery set and a validation set, and we did a combination of different techniques using whole genome, including whole genome sequencing, whole exome sequencing, that's the coding region sequencing, and then RNA sequencing, and so on. So one of the first things that we found, I don't know, many of you may or may not be familiar with these. This is called a Surcos plot. And essentially what it does, it summarizes the findings of all those billions of reads into one slide, into one cartoon. So this is a typical alveolar rhabdomyosarcoma. And I say low aneuploidy because this gray bar here tells you the copy number of the genome across all the chromosomes. You can see that there is a little bit of aneuploidy, but not a significant aneuploidy in terms of different copy numbers in different parts of the genome. There is low somatic mutation burden. What I mean by that is if there was a mutation in the tumor and not in the DNA, one would see the genes on the outside labeled in either as a single nucleotide variant or insertion deletion. And you can see in this rhabdomyosarcoma that has the 213 translocation here doesn't have any somatic mutation. Right? So a very aggressive cancer with very few mutations is what is happening in these fusion-positive rhabdos. Whereas in fusion-negative rhabdos, you can see there's a lot more aneuploidy, different copy number alterations. You see a lot more mutations. In this case, I'm showing you an NRAS mutation that occurs, as well as chromosomal gains and losses shown here. So this is fusion-negative rhabdos have a higher degree of aneuploidy and a higher somatic burden. So this is the total number of somatic mutations per tumor in embryonal and this is still about 3 times less than in adult cancers. Whereas, if you remember previously, in the tumors that have the fusion genes, it's around 5 or 6 or so mutations, the median number of mutations, whereas it's 18 in this case, but it's still lower than adult cancers. Now, if you take all that data and then summarize it in these kind of heat maps that you also may see in some of the literature, what we found, you can see there's a big difference between these fusion-positive rhabdos and fusion-negative rhabdos, where the fusion-negative rhabdos primarily have mutations in the RAS pathway. So here's NRAS, KRAS, HRAS, and so on. They also, as these cancers, have amplifications and deletions of genes that are potential targets shown here. And if you want to know more detail, you can look up this paper. But what I show in arrow here is that many of these, certainly in rhabdomyosarcoma, particularly in fusion-negative rhabdos, are potentially targetable in terms of therapeutics. And so this allows actually ability to do more precision therapy type of trials that many centers are interested in. But what you noticed here is on the right, can you see there's a silent zone? In other words, there's no obvious recurrent somatic mutation in these fusion-negative rhabdos. So the question comes, what are actually the drivers in these apparently silent fusion-negative rhabdomyosarcomas where there's no obvious drivers, mutations? The other thing just to note, you can see there's very few mutations, somatic single nucleotide variants actually within the fusion-positive rhabdos, indicating that the fusion gene itself is the main driver. So to answer the question, what's within this silent zone, what we turned to is combining RNA sequencing with DNA sequencing, and then looked at how many of these genes are actually expressed and what is the level of expression. So the size of the circle tells the level of expression, and the color tells you which the variant allele frequency. In other words, if the mutant allele is expressed and how much of it is expressed. And what you can see here, that in many of these cases, many of these are druggable genes. They're expressed in the genome, and some of the drivers are very high level of expression, and in many cases the variant allele frequency is actually close to 1. In other words, both alleles are mutated. So that increases the dose of the oncogene essentially. Now if you take all the data and then see what are the pathways that are altered by these genes that are mutated in these multiple cancers, you can see that essentially in rhabdomyosarcoma it's the FGFR RAS signaling pathway that's mutated in these cancers. And what it also tells you is that because it tells you there's a lot of redundancy within the protein-protein network, such that it's and we've all seen this in cases where we start using a single agent to inhibit one gene, for example FGFR4, it's most likely that the other proteins will actually compensate, and therefore, just like in bacteria, the potential for resistance is there. And so this is the reason why combination therapies is necessary, not just with chemotherapy, but also in some of these precision therapy trials targeting these small these proteins. Oops. So in summary, what I showed you for this part of the talk is that the importance of doing these integrated multi-dimensional omics methods to look for precision therapy targets for therapies. And then what I also showed you is that in these alveolar rhabdos, there's very few mutations, and the fusion gene, unfortunately, because these are hard to target, but they are the main oncogenic driver, just like in Ewing sarcoma. That's the primary, the fusion gene is the primary driver. And then, but interestingly, in rhabdomyosarcoma, in fusion-negative rhabdos, there certainly is a lot of targets that are potential therapeutic targets, and so that is the subject of much of our research that we're going on now, which is looking for targeting these rhabdomyosarcomas and the fusion gene itself. So I'm going to talk to you a little bit in the next part of the talk and some of the challenges and opportunities. So the genomics is pretty much done to a large degree in terms of many of the tumors have been sequenced in pediatrics and in adults. So what are the challenges and opportunities in pediatric cancers in this post-genomic world that we live in? So this, again, I'm going to keep coming back to this, and you've already heard about osteosarcoma, rhabdosarcomas, and Ewing sarcomas, and neuroblastoma is another. So metastatic disease essentially remains incurable. So this led us, again, I showed you how in pediatric cancers, the number of somatic mutations per cancer is very low compared to the mutational burden in adult cancers. So much of these studies have been done actually in diagnostic samples. So all the sequencing studies that you've seen in manuscripts have been done on cancers that were initially at presentation, so before any therapy was given. In that situation, the mutational burden is certainly low compared to adult cancers. So this led us to a study where we wanted to use, again, multidimensional clinical genomics to look to investigate a series of patients that actually came to the NCI. And these are, at the NCI, we see adolescent young adults and patients with relapsed and refractory cancers. And I'm going to just show you the results of this study that was recently published. So since from 2020 to 2014, we enrolled on this omics trial 59 patients with essentially with relapse cancers. And so because of this represents the type of cancers that we actually see within the NCI. The age range was 7 months to 25 years. And essentially what we did, we profiled using exome, which is sequencing the protein coding genes, the germline and tumor. We did RNA-seq of the tumor. We did SNP arrays of the tumor itself. And the purpose of this study was to find what's called actionable mutations. And I'm going to just define those two just so that you know what I'm referring to as actionable. So an actionable germline mutation essentially is a loss of function mutation of a cancer gene or a pathogenic or likely pathogenic mutation of a set of genes that the American College of Medical Genetics has said that actually if you see a mutation in this gene, it is of significance in terms of the management of the patient, not necessarily for cancer, but maybe for some other diseases. So this is a definition of actionable germline mutation. And actionable somatic mutations, essentially, we define for this manuscript as genomic alterations that changes the patient's diagnosis or may be targeted with an FDA-approved drug or in the context of existing clinical trial according to, there's like a matched trial in adult cancers where patients with cancer are being matched to a drug or a trial based on the mutations that's identified. So we use similar criteria to see what happens in pediatrics, basically. And so I'm not expecting to read this all, but essentially, when we sequenced all these tumors, this was really surprising to us, but we find that 50% of all our patients with cancer actually had an actionable mutation. And I've just shown you some of the cancers, the diagnosis, the mutations that we identified, and actually the drug that can potentially be used in clinical trials, and the match criteria, so which you can't read, but you can look in the paper. But essentially, 50% of pediatric cancers that came to the NCI had an actionable mutation. And then the other surprising thing to us was many of us who are oncologists don't think about this, but actually the question is how many of these patients actually have actionable germline mutations, so mutations that would alter the management of the patient and the family. And what did we find? We found that many of these cancers had mutations in loss of function mutations in these actual driver genes that actually may potentially have serious consequences to the management of this patient or the family. Again, more detail you can get from this paper here. So I think hopefully I showed you that the importance of actually doing these types of multi-dimensional genomics trial assays in the clinical setting, and that this can be done in real time. So there are many groups who are attempting this in many different hospitals. So this 50% of children and adults with cancers have these potentially actionable mutations is very promising in terms of clinical trials. And I already mentioned to you about 10% have these actionable germline mutations. And what we also find, interestingly, is some of these germline mutations in genes are actually actionable. So actually, even though it's in the germline, it's also present in the tumor, and these can actually be therapeutically targeted in the patient. And there are examples already of this in terms of RET mutations and trials that Brigitte Wiedemann is doing at the NCI. And you may or may not know, but the Children's Oncology Group is about to launch a pediatric MATCH trial, which is essentially going to be using some of the genomics methods to direct therapy in the setting of relapse cancers. So this just is a complicated diagram. I'm not going to go over it too much, but essentially this is within the Center for Cancer Research of the NCI. Patients will be enrolled on the sequencing trials where the sequencing results will be actually directly reportable through the electronic medical record to the physicians, as well as as well as potentially developing precision therapy trials. So this is a platform that is currently being developed with a very rapid turnaround. So the last part of my talk, I'm going to just talk to you, go back to the theme of why single agent therapy, even if you find an actionable driver, it's not a slam dunk. In other words, so this is a figure from a very famous publication back in 2011 of a patient with a BRAF mutation, V600E, melanoma, and you can see that within 15 weeks treatment of a very specific inhibitor of this mutation, there was an almost complete response and a dramatic response. But then within 23 weeks of this therapy, it comes back, and often in the majority of cases, it's the exact same tumor. You can almost put one picture on top of the other and get exactly the same tumors come back. So that tells you that these cancers are rapidly versatile, and there is the potential for rapid resistance if you use single agents. So that led us in our group to move on to develop immune therapeutics for rhabdomyosarcoma. And we have three projects that we're doing essentially by three different people. Bhaskar here is actually developing antibody drug conjugate against cell surface proteins. And then Nitya here, she's a graduate student from Georgetown, is working on chimeric antigen receptor therapies against rhabdomyosarcoma. And just a new postdoc, Martin here, is actually developing bispecifics, again, in rhabdomyosarcoma. And this is part of we're part of a Stand Up to Cancer St. Baldrick's Immunogenomics Dream Team that actually is doing multiple different targets for cancer. So just a few slides why we're interested in this. So I've already mentioned to you this very important center of the universe gene called FGFR4, which is we think is an attractive immunotherapeutic target. I told you already that it's on the cell surface, so it makes it targetable, and it's very highly expressed in rhabdos. I've already mentioned to you that it's an oncogene, it's driven by a fusion gene, and it's also interestingly overexpressed in many different cancer types. And I've just listed a few: lung, liver, breast, pancreas, and prostate has high expression. So we've gone ahead, and made multiple monoclonal antibodies, both rabbit, mouse, and human, against FGFR4. And essentially what we found was that in many of these antibodies, you can't really see it here, but once it binds, I'll give you one example here. When it binds onto the rhabdomyosarcoma cell line, it gets rapidly internalized, and actually makes it promising as a antibody that can actually conjugate drugs to. Trojan horse, that's a good name. So what we find here, so we actually have I've developed some antibody drug conjugate, and when we tested them in normal muscle cell lines, there is minimal in terms of inhibition of growth. But what's really exciting is that when we now test alveolar rhabdomyosarcoma cell line, there's very specific inhibition of growth in these cancers, indicating that this is going to be very promising as a therapeutic in rhabdomyosarcoma. So then Nitya has actually made significant progress in actually making CAR T cells, again using FGFR4 as a target. And here we're comparing essentially rhabdomyosarcoma cell line versus the Raji cell line, which doesn't express FGFR4 but expresses CD22. And you can see that CAR T cells against FGFR4 is very specific. These are different binders essentially. So we have very effective killing of a rhabdomyosarcoma cell line when you use CAR T cells against FGFR4. And this is just to control that we can actually kill Raji cells with CD22 CARs. So this is actually in collaboration with Lentigen and Miltenyi, and Rima Sorrentis has been instrumental actually working with us to actually hopefully actually get this in a clinical trial. So in conclusion, hopefully I did a rapid tour showing you that there are targets, at least small molecule targets, in the majority of rhabdomyosarcomas, and this is one area that we're working on. And I haven't shown any work on that, but we certainly screening multiple small molecules for therapeutics in rhabdos. I showed you that pediatric cancers have a very low somatic burden at diagnosis. What I didn't show you, but in that manuscript, what we found was that the number of mutations in pediatric cancers actually increases to almost adult levels at relapse. So as cells divide, they continue to accumulate mutations, which makes it promising actually as more therapeutic targets may actually evolve, which is bad for the patient, but actually there may be potential therapeutics in the future. I mentioned to you the importance of germline mutations, and I think in the future, I think it is really important to actually sequence every pediatric cancer, the germline, to look for potential germline mutations. I mentioned to you about how single therapies are not, even against actionable mutations, are going to be unlikely to be curative, and therefore the need for combination therapies. I mentioned to you some of our plans for precision-based small molecule therapeutics within the Center for Cancer Research. And finally, I mentioned to you how we are bringing forward FGFR4 as a therapeutic target, both small molecules and immunotherapeutics. And I think this is going to be very promising for these rhabdomyosarcomas in the future. So this is obviously a huge amount of work done by a huge number of people, and I mentioned to you already Bhaskar and Nitya and Martin. June heads is a biological, he's our staff scientist and heads my lab. There's a bioinformatics group that was working on these projects, and we have had a lot of support from the NCI leadership. The immunogenomics dream team, the Son of the Cancer, St. Baldrick's dream team, these are some of the members within the team, and this is led by John Maris and Crystal Maffel, who used to be at the Pediatric Oncology branch. A lot of support from our pathologists and collaborations with other groups, and I already mentioned to you a very good collaboration with Lentigen and Miltenyi, allowing us to take FGFR4 CAR T cells to the clinic, we hope. And I think, and also just other investigators within the NCI and elsewhere in terms of supplying tumors and so on. So I think that's it, and this is the Stand Up to Cancer Dream Team, and you can see that this is a very exciting collaboration that's ongoing, the work is ongoing. So thank you very much. I'm so sorry it's so rapid, and I'll be happy to take questions.
Yeah. All right. Thanks a lot, Javed. We have time for a couple of questions. The other Javed.
Thank you for a great talk. Are there any efforts to include proteomics into the clinomics and the multidimensional analysis?
Yeah.
Because it seems like for some of your cohorts that don't have any genomic mutation, post-translation modification would probably be an answer.
Yeah. Well, first of all, I wanted to meet my namesake for a long time. There is. So we're doing phosphoproteomics in some of these cancers, but we found that the need for fresh tissue is more important in terms of doing phosphoproteomics. Certainly in cell line work, we're doing that. But also, I think you made a lot more advancements than we have. But in terms of closeness to the clinic, it's not as close as genomics. But I'm sure people are. We're investigating it, but it hasn't gone to the far as a test that can be used to actually make therapeutic decisions yet. It's just in the research stage at the moment, certainly in our lab anyway.
Other questions?
You commented that frequently tumors can evade a targeted therapy because of other connections in the pathway that can be activated. I'm curious, in the case of FGFR4, when you disrupt that with a small molecule, I presume there are concerns, like you indicated on the slide, there could be FGFs that are upregulated. But from the immune therapy approach, is there any evidence at this point that if you target it with a, say, chimeric T-cell receptor, would you expect a change over time in the profile of cells that express FGFR4 at high levels versus low levels? Or do you think that's that characteristic is likely to persist, and so the immune therapy approach might be more successful because of that?
It's a great question, and for example, in the NCI and others, the CD19 CAR therapy in leukemia, there is a rapid, in some cases, patients relapse with CD19 negative leukemia. And in those cases, essentially, either you get a mutation in the epitope, or actually, some of those cells actually are not dependent on CD19 for survival. So I think even with immune therapeutics, resistance is going to develop a single agent. And it's going to be a battle of killing the last cell versus resistance development. FGFR4 in alveolar rhabdos is a direct target. In other words, Pax3 sits on the enhancer region and activates it. And it's necessary for survival. But I'm sure IGF-1R or other genes will take over. And so the question is still out in terms of, the hope is that you kill the last cell before the emergence of resistance. But I suspect resistance will develop.
So, Javed, really an amazing overview, and I hope people got an appreciation for just the number of different technologies that are necessary to approach the complexity of all the different aspects of the tumors is really impressive. In your assessment that you found 50% actionable mutations, with Kim Stegmeyer's study out of the Broad, as you know, it was less than 10%, maybe 7, maybe 9%. What do you ascribe to the differences of how you found 50% and she only found less than 10%?
So I would say several things. One is that your definition of actionable. So if you say, for example, MYC-N, where there isn't an exact drug that inhibits MYC-N, but there are BRD inhibitors downstream, and these cells are exquisitely sensitive to downstream inhibitors. So we use the match criteria, which isn't necessarily one mutation, one drug. It's one mutation and downstream pathways and one drug. That's one. I'm sure there's selection bias because at the NCI we see very specific cancers that multiply relapse. And I suspect in a much larger study, the numbers have dropped, probably not as high as 50%. But many of the 3 other studies that have been published in a similar small studies, they've had similar numbers. But again, I think it's selection bias is part of it.
Yeah, I think your point about how you define an actionable target is critical here, because if you make it really narrow, single drug, single target, ready to give tomorrow, versus single drug, maybe there's a pathway that could be, that something in that pathway, and then possibly ready soon, you have a very wide range. Yeah, fantastic. Well, I would like to thank all of the morning speakers, the first session.